Hi, everyone. Welcome to our Tuesday get-together on the cerebellum. Today, I'm really happy to tell you about Gabriel Stine. I met Gabriel at SFN few months ago, and I was just blown away by the work that he had done in Mike Shadlin's lab. And he just told me that the paper that he's going to be presenting was just accepted at Neurons. So you're getting a preview of really wonderful work. Gabriel, thank you so much for being with us, and I'll let you take over.
Great. Well, thanks, Reza, for inviting me and everyone for coming. So a big disclaimer: the talk is not particularly relevant, or is not on the cerebellum. So I know it's a cerebellum seminar, but hopefully you all still find it interesting and can take some things away from it. So I'm actually now a postdoc in Mehrdad Jazaeri's lab, and I'm working on a project that's looking at the role of the dentate nucleus in non-motor learning. So I'm excited to share my PhD work with all of you, but I'm also happy to sort of introduce myself to the cerebellum world. So yeah, I'll tell you about my PhD work on the neurobiology of decision making. And I want you to imagine that you're at Wimbledon playing Rafael Nadal, and you're standing at net, and Nadal slams a forehand, and the ball's coming right towards you. And you need to make a decision about whether to move to the left and hit a forehand volley, or move to the right and hit a backhand volley. And critically, not only do you need to make the right decision, but you need to make it in the right amount of time, or else something like this might happen. So I want you to get a sense that there's two stages to this process. You have to deliberate about what to do, and you have to commit to a proposition or an action. And this transition from deliberation to commitment is fundamental. It's something we're doing all the time, and we'd like to know how the brain does it. So to get at this question, we study decision-making in macaque monkeys. And all of the data I'll be telling you about today comes from two monkeys performing a random dot motion discrimination task. In this task, a monkey looks at a computer screen. A cloud of randomly moving dots appears, which move to the right or to the left. And whenever the monkey's ready, it indicates whether it thinks it's moving to the right or to the left with a corresponding eye movement. And critically, this is what we call a reaction time, or a free response paradigm, which means that, like I mentioned, the monkey decides when it responds. There's no go cue, nothing like that. And this is going to be really important because this reaction time paradigm identifies the transition from deliberation to commitment on each decision. Now, algorithmically, we have a pretty good idea of how these types of decisions are achieved, which is through a process of bounded evidence accumulation. So I'll show you an example of this model in action for a rightward decision. And what this model says is that information from the stimulus, or sensory input from the stimulus, is transduced into noisy momentary evidence, here represented as a Gaussian. And then this noisy momentary evidence is sampled over time and integrated until a rightward or leftward threshold is exceeded. And here I'm depicting this as a race between two accumulators, one for a rightward choice, one for a leftward choice. And this particular trial ended in a rightward choice because that's the accumulator that reached its threshold first. And these bounded evidence accumulation models explain a lot of observations that we see in decision making. The sort of classic example is the fact that harder decisions take longer to make, and this type of model explains that with a common mechanism. In the brain, people have found representations of this evidence accumulation process in a number of areas, but it's probably been most studied in the lateral intraparietal area. And LIP has spatially selective persistent activity. And what Mike Shadlin and his colleagues found over 20 years ago now is that if you place one of the choice targets that the monkey uses to report its decision within the response field of an LIP neuron, that these neurons represent the accumulation of evidence. And so here I'm showing you the average over many neurons and many decisions split for different motion strengths and directions. And what they found is that activity builds up or down at a rate that depends on the motion strength and direction. And that's exactly what you'd expect for a representation of accumulated evidence. So here's just a few example motion strengths. Here are the in-betweens. And I want to make a really important point, which is going to be important for the rest of the talk, which is that these are averages over many neurons and over many decisions. It's sort of historically has been necessary to do this because we are only recording one or a handful of neurons at a time. And I'm just going to highlight on one of these averages, right? And this is the mean, again, over many trials. And critically, this is not what we think is occurring on any single decision, right? On any single decision, we think what's going on is a drift diffusion process. And by averaging across many decisions, we get to see the drift or the hypothetical drift because that's common to every decision. It's deterministic. But what we lose is the diffusion component of this because the diffusion component is unique on each trial. So when we average, we only see the black line, we see the drift, but we hide or we lose the dynamics that are unique to each decision. And that'll be important going forward. What Mike and his colleagues also found is that if you align activity to the time of the saccade, right, the time that the monkey is committing to a decision, they found that activity, regardless of how long the monkey takes to make its decision, activity coalesces to a common firing rate about 80 milliseconds before the saccade. And this has long been taken as a sign that some downstream area is applying a threshold to LIP activity or activity that's correlated with LIP activity. But exactly how this threshold works is not really known, and we'll go into that more as well. Now, finally, LIP doesn't act alone. It's highly interconnected with a number of brain areas, and here is a simplified schematic of that. And my old cortical chauvinism is on full display here because I have not included the cerebellum, and this figure will definitely get updated in the future. But LIP, right, is connected to a number of brain regions. Its two main projections are to the frontal eye fields and the superior colliculus, or SC. And as you probably guessed from the title, my talk will focus on its projection to the superior colliculus. And ultimately, we'd like to understand how all of these areas interact and cooperate to form and commit to a decision. Now, like I said, LIP and SC are connected to each other. And when you just look at their activity during a simple delayed saccade task, they look pretty similar. So here's data from Pare and Wirtz of activity in a delayed saccade task. And it's qualitatively very similar. And the main difference shown here is that SC has a large saccadic burst of activity. Others have recorded in superior colliculus during decision-making. So here's data from Horowitz and Newsom, right? And they found sort of qualitatively similar activity profiles to what we've seen in LIP. And more recent work from Michel Basso's lab has also found qualitatively similar representations, though they also found some interesting differences as well. And what these data have led to the idea in the field that the accumulation of evidence is this distributed process across many brain regions, but the idea has not really been tested strongly because people haven't recorded in the same animals in the same task and at the same time. And here I'll show you some evidence that things are a bit more modular than what we actually appreciated. Right, and of course, others have recorded from other regions. Ding and Gold specifically is recorded from the FEF and the caudate nucleus, and also found similar representations to what we see in LIP. So in my talk, I hope to answer at least two central questions. The first one is decision-related activity in SC similar to decision-related activity in LIP? And the second one is how do these two areas interact during decisions? And I'll say also, I forgot to mention this in the beginning, that please interrupt if anything is unclear or if any questions, be happy to answer. So to answer these two questions, we simultaneously recorded in LIP and in SC. And in SC, we used 32-channel V probes, sort of what was the standard. And in LIP, we used these new prototype macaque neuropixels probes, which I'll explain in more detail in just a second. So in SC, it has a retinotopic map, as many of you probably know. And so because we can approach it normal to that map on this 32-channel probe, essentially all of the neurons have the same response field. It might look something like this. So here's a heat map depicting the firing rate of an example SC neuron during a delayed saccade test. So those white points are different locations that the animal made saccades to. And recording an LIP at the same time is really not enough. Ideally, what we want is neurons in LIP that have the same response field as the SC neurons, because that's how we know that the two groups of neurons are actually connected or talk to each other. Right, so ideally we would have LIP neurons that have response fields that look something like this. And historically, this has been really difficult. If you put a V probe in LIP, you get a few dozen LIP neurons, and the chance of just happening to find an LIP neuron that has a response field in the same place as the SC neurons is really low. And that's because LIP does not have strong retinotopic organization. But now with these neuropixels probes, doing this becomes much more plausible. So these probes are 45 millimeters long. They have 384 active sites at any time. They're actually now available just as a few weeks ago. So that's extremely exciting. And in any given session, we got about 80 to 200 neurons on the probe that we could record simultaneously. And of those, only about 5 to 40 of them had a response field that overlapped the target and the SC neurons, what we call T-in neurons, or target in neurons. Right, so 5 to 40 per session. And just to give you an idea, I did months and months of experiments using V probes instead of neuropixels probes, and I would get 0 to 1 neurons per session. So this type of thing was implausible before, and I just want to give you an idea of the not only do you get more neurons with these neuropixels probes, but just the space of possible experiments is just so much larger now, and I think that's super exciting for the field.
Hi, Gabriel. I have a doubt here. The plots of firing rate you shown, these are at what time of stimulus or saccades?
So this is during the delay period when the monkey knows where it's going to make an eye movement, but is waiting to get a go cue.
Okay, so it's averaged over the delay period.
I'm sorry, maybe this is a delayed saccade test. So this is not during the decision-making task. This is just the monkey's given a target, and once the fixation point is extinguished, it makes an eye movement. Does that answer your question?
Yeah, and also I had another doubt that the activity in LIP integrates over time in a decision-making task, like a drift-diffusion model. So similar activity has been observed in SC also. Have I missed that point, like in your last slide?
Okay, yeah.
Similar accumulation of activity.
So we've find activity in, it's a bit nuanced. So we find activity in LIP that's consistent with the drift diffusion process, but because we're averaging over many trials, we actually don't get to see the diffusion dynamics. So it's always been an inference from the averages over trials. And when you average over trials in SC, things look qualitatively similar. But we'll get to this. I think I'll answer your question in just a few slides.
Okay. Thank you.
Right, so now with NeuroPixels probes, we can record from populations of neurons in the two areas that have matched spatial response fields. So here's what the data look like. This is an example session from 17 LIP neurons that share the same response field, averaged over trials. So it's very similar to the plot I showed you before. And you know, they've replicated years of previous work where activity builds up or down at a rate that depends on the motion strength and direction. And like I mentioned and just mentioned, you know, we've always hypothesized that these averages are comprised of these drift diffusion paths. So this is like a simulated diffusion process, what we imagine is happening on single trials. And now that we have a population of neurons recorded simultaneously, we have the ability to go and look at what's going on in single trials. And simply what we're going to do is just take these 17 neurons and average them together and look at the average of the 17 neurons on a single decision. So that's, you know, here's one example decision. You just get, you know, a wiggly line. This is the average. So this is one decision, and this is what it looks like over many decisions. And I hope you appreciate that just visually, these two look very similar. So we've always inferred the thing on the left, and now with a population, we can directly observe what looks like drift diffusion dynamics on single trials. Additionally, we can align things to the time of the saccade, and like previous work, we found that activity coalesces to a common firing rate. And again, we can sort of zoom in and look at one of these curves and ask what does the single trials look like, and here's that. And so things coalesce on the left. I showed you that things coalesce across motion strengths, and here on the right, I'm showing you that within a single motion strength, things reach a point of minimum variance just before the decision. So again, we've sort of had a lot of ways of inferring that this was all true using the data we had in the past, but this is the first time we're actually able to just look at the single trials and verify that it was correct. So then, you know, we wanted to know what is SC doing and how does it compare? So on the left, I'm showing you the trial averaged activity in SC, and I think qualitatively it looks very similar. Activity builds up and down at a rate that depends on the motion strength and direction. And again, the main difference in SC is this large saccadic burst of activity. It goes off the charts. It's so big. But things took a turn when we looked at the single trials, right? So now we can do the same exact analysis. We can look at the average of the SC population on single trials, right? The saccadic burst. And on single trials, things look very different. So like we saw in the trial averages, we see this large saccadic burst in the single trials. But what we see here and what we didn't see in the trial averages are these non-saccadic bursts of activity. And these are bursts that look a lot like the saccadic bursts, but for whatever reason are not associated with the saccade. And they're not aligned to any particular trial event. They sort of happen at seemingly random times. And what that means is that when we average over many trials, they get averaged out. So you don't see them in the trial averages, but they're very clear in the single trial activity. So this was surprising to us, right? And I want to just hammer this point that we found similar trial averaged activity, but distinct single trial dynamics. And I think that's maybe a cautionary tale for what most people in systems neuroscience are doing, that things can be hidden by averaging over trials. And so the fact that the single trial dynamics were different suggested to us that maybe the two areas are performing different computations. So, you know, LIP is accumulating evidence and maybe SC is doing something else. But we wanted to test this distinct computation idea more strongly. And so to do that, we went in and we injected really small, subtle, and brief motion pulses into the stimulus, right? So these last 100 milliseconds, and they're really subtle. You can't really detect them with the naked eye. but they do serve to slightly bias the animal's choices. So here on the top, I'm showing you reaction times as a function of motion strength. On the bottom is choices as a function of motion strength, or proportion rightward choices. And the two curves are separated by whether there was a rightward pulse in the trial or a leftward pulse. So the effect is very subtle, which was by design, but reliable. And what we could then do is align activity in the two areas to the time of the pulse and look at what is essentially impulse response function, right? And if an area is integrating evidence over time, then a brief pulse of evidence should lead to a persistent change in the firing rate. And that's what I'm showing you here is basically a regression weight, the effect of the pulse on activity in LIP, which has a persistent effect on LIP activity, consistent with temporal integration. And in SC, the pulse also had an effect, but it had a much more transient effect on SC activity. So again, this tells us that maybe this is all consistent with the idea that SC is not accumulating evidence over time, or at least if it was doing it on a shorter time scale. So that evidence combined with these distinct single trial dynamics, tells us that these two areas are doing different things during decisions. And we were particularly intrigued by these bursts, these non-saccadic bursts of activity. And we wanted to know, is there something in LIP that is correlated or maybe triggering these bursts, which might tell us something about what computation SC is performing. So to investigate this more, we developed an algorithm that identified these bursts and which allowed us to align them in time. And then we simply took the average of those. So here's the mean non-saccadic burst in SC as a function of time. And then we simply asked, what is LIP activity doing at this same time point? Because we recorded simultaneously. And so here's the average activity in LIP aligned to the time of the burst in SC. And what we found is this sort of what I'm going to call an uptick in activity in LIP at the time of the burst, a sort of increase in activity, which then leads to sort of a new higher firing rate. Now, this uptick is highly predictive of whether a non-saccadic burst occurs in SC. So here I'm showing you the probability of observing a non-saccadic burst in SC as a function of the uptick size in LIP. We found a very close quantitative relationship between the two. So we then did the same exact thing for the saccadic burst. So here's the mean saccadic burst in SC, right? We have our algorithm, the same algorithm. And then we can ask what happens in LIP at the time of the saccadic burst. And again, we found a very similar looking signal, this uptick in activity. And in fact, if people are familiar with LIP, people have often found what they call perisaccadic burst in LIP activity at the time of the eye movement. LIP has, this sort of small burst or uptick in activity. And this is exactly that. It's just now aligned to the saccadic burst. But the main difference between the uptick in LIP during saccades and during the non-saccadic burst is that the upticks that occurred during the saccadic burst stemmed from a higher baseline of activity. So this led us to the idea that maybe what's going on is that an uptick is sort of tickling the bursting mechanism in SC, and that the saccadic burst occurs when that uptick occurs or stems from a high firing rate. And if you think about, if we are hypothesizing that these bursts in SC are a sign of a threshold crossing, I just want to plant the seed, is that it's exactly what you would want, is that you always have to cross a threshold from below. So you basically always need a positive derivative event to cross the threshold from below. And so it's maybe not that surprising that we find an uptick in activity during the saccadic burst. The fact that they also occur during the non-saccadic burst is telling us that this might be something important to the bursting mechanism in Colliculus. With all that said, this is all completely correlational, right? So it could be the case that the SC bursts are causing the upticks in LAP. And so this is just a hypothesis, and more on that in just a second. So going back to our central questions, we first wanted to know, is decision-related activity in SC similar to that in LIP? And I showed you that they're qualitatively different, and that LIP reflects drift-diffusion dynamics and SC reflects bursting dynamics. Then we wanted to know, how do LIP and SC interact? And I showed you that bursts in SC are correlated with sharp upticks in LIP activity. So I hinted at this, but it led us to the hypothesis that SC is implementing the decision threshold. So, right, if it's not accumulating evidence, maybe it's implementing the decision threshold. And the crossing of this threshold manifests as a burst, which generates the eye movements and terminates the integration process. And so going back to the accumulation model, right, we think LIP is representing this accumulation of evidence signal, the decision variable, right? And maybe we can place SC on the schematic as the threshold. Any questions before I move on to the next section? Okay, so to test this hypothesis, we wanted to do a causal experiment. And so to do that, we unilaterally injected mucimol into SC with very small volumes, and we simultaneously recorded an LIP with these new neuropixels probes. And so many have inactivated SC during saccade tasks. And when you do this, you don't abolish the monkey's ability to make eye movements. You simply slow them down in a confined region of space. So here I'm showing you a heat map of the monkey's saccadic latency or saccadic reaction time as a functional location after SC inactivation. And so what you should see is that in just sort of the left hemifield, in that confined region, the monkey is about 50 milliseconds slower at initiating a saccade. This is in a delayed saccade task. And in this confined region of space, we're going to call the inactivation field, or the inactivated region. And we're going to place the choice target, like we did before, within the inactivated region. So it's worth walking through our hypothesis and exactly what we think inactivating SC is going to do to the monkey's behavior. So if our hypothesis is that inactivating SC is going to lead to a deficit in decision termination, here's what we would expect. So we have our two accumulators, right, for left and right. And here in this case, we're injecting mucimol into the right SC. And so what we hypothesize is that that is going to increase the decision threshold for making a leftward choice. And by raising that decision threshold, what that means is that more evidence needs to be accumulated to reach that same threshold. Now, if we simulate this process or this model and look at the behavior, this is what we'd expect. So here I'm showing you again the reaction times at the top and the proportion rightward choices on the bottom. It's a split by whether it's before or and after the simulated inactivation. And we should expect three observations in the behavioral data. The first is we should see a choice bias. The monkey should be less likely to make a leftward choice in this case. When the monkey does make that leftward choice, we should see an increase in reaction time. And third, and most importantly, we should actually observe a small increase in the slope of the psychometric function. And the intuition behind that is by raising the decision threshold. When the monkey does make that decision, it's doing so based on more accumulated evidence than before. And that leads to slightly better performance or a slightly steeper slope of the psychometric function. Right, and that's a sort of counterintuitive prediction, because usually when you think about inactivating a brain region, you think about decreasing the slope or decreasing performance, but we're making the opposite prediction. So the null hypothesis, or an alternative hypothesis, is that inactivating SC is simply going to cause a deficit in motor production. We're just going to make the monkey a little slower at initiating its eye movement, but the decision threshold was maybe say upstream of SC. And so you're just slowing things down. And so the idea there is that in this model, you might imagine that there's some non-decision time, where it takes some amount of time to initiate the movement, has not, sort of orthogonal to the decision itself. And by inactivating SC, we're simply increasing that delay. And what this would predict, if we simulate this, is that we should just simply delay, reaction time should be longer for leftward choices, and there should be no effect on the monkey's choices. So here I'm showing you again just that simulated effect of increasing the decision threshold. And here's what we got after we injected mucimol unilaterally. And just visually, we saw a very clear correspondence between those two. We'll dive into it more in just a second. And this is what we get after saline injection. So going back to our 3 predictions, right? We first expected to see a choice bias, an ipsilateral choice bias, and that's exactly what we found. The green curve is shifted to the left of the black. We found an increase in reaction time. Here I'm showing you the change in reaction time as a function of motion strength. And on the right, that dashed line on the bottom, this is the change in reaction time in delayed saccade test. So our increase in reaction time in the decision task is much larger than what you'd expect from just a simple increase in saccadic latency. And third, and most importantly, it's really hard to see in the plot, but we actually found a significant increase in the slope of the psychometric function. So you might note, so here I'm showing you the slope before and after either mucimol or saline. You might notice that the monkey generally does better in the second half of the session, and that's not too uncommon for our lab, but that the increase in slope after mucimol is significantly larger than what you'd expect from saline. And this increase in slope, this slope effect, was highly consistent across sessions. We saw it in every single session except one. And so we were sort of impressed by this result because it's relatively easy to cause bias. It's relatively easy to increase reaction times, but it's pretty hard. There's relatively few mechanisms or few manipulations that would actually increase the slope of the psychometric function. So we thought this was compelling evidence that the effect of SC inactivation on behavior is at least consistent with an increase in decision threshold. So I mentioned that in this experiment, we recorded in an LIP with a neuropixels probe at the same time that we did this in about half the sessions. And again, what the neuropixels recordings does, the large yield, allows us to find LIP neurons that have a response field that are within the inactivated region. And we wanted to know, does the increase in the decision threshold manifests itself in LIP activity, or in what way does it manifest itself in LIP activity? And again, it's worth walking through our hypothesis. So if we if we think that inactivating SC is increasing the threshold for making a leftward choice, such that you need to accumulate more evidence to reach that choice, if LIP is representing that accumulated evidence, then what we would predict is that if we look at LIP activity at the time of decision termination, at the time of the saccade, LIP should reach a higher level of activity. Really simple prediction. And so here's averaged LIP activity aligned to the saccade before SC inactivation, and here's what we got after SC inactivation. So just like we predicted, we found a pretty substantial increase in firing rate after SC inactivation in LIP, as if this was the threshold before SC inactivation, it's as if the threshold was raised to a higher level. So we thought this was really nice. We sort of have this, we're able to confirm our hypothesis using a simultaneously recorded activity in LIP. And this increase in firing rate was also consistent across neurons. So here I'm showing you firing rate before SC inactivation and after SC inactivation for each neuron. So there's one last thing I want to point out to you, and that was intriguing to us, which is has to do with the upticks I mentioned before. right? And I mentioned it was all correlational, the relationship between upticks and bursts, and we'd like some causal evidence. And right, and here's the plot I showed you before, just showing that this uptick in LIP occurs at the time of the saccadic burst. And right, if you're looking at activity in LIP at the time of saccade before SC inactivation, we see a similar profile. But if you're an astute observer, you might notice that this uptick seems to be gone after SC inactivation, right? This green curve is just sort of gradually increasing. It doesn't have this change in derivative. And this is very intriguing to us. And I thought, well, okay, maybe, what happened to the uptick? Maybe it was caused by bursting in SC. And so when we inactivate SC, the uptick no longer occurs. So that's one possibility. The other possibility is that the uptake is still occurring in LIP, but it's just simply no longer aligned, or the alignment is less clear with the saccade or with decision termination, such that when you average across trials, it gets smeared over time. And so we wanted to think of a way to test this. And here I'm showing you a heat map of every single trial in this data set, split by before or after SC inactivation. And I'm plotting the derivative of the firing rate, or the change in firing rate as a function of time. What I want you to get a sense of is that before SC inactivation, we have these sort of streaks of greenish yellow that are sort of stacked right before the saccade. And after SC inactivation, it's less clear. Maybe there's something there, maybe not. So if the idea is that these upticks are simply no longer aligned with the saccade, then it should be possible to find a realignment of the data that now makes them visible in the average. And so we exploited this algorithm developed by Williams et al. This is a neuron paper where they develop a time shifting and time warping algorithm to align events that occur in neural data. And we use the simplest version of this algorithm, which is a simple time shift. So on each trial, we're just going to find we're just going to move the x-axis around to align features in the data. And we're going to do this separately for data before and after SC inactivation. So when we do this trial-wise time shift, this is what we get. And we get things that look qualitatively similar before and after SC inactivation. Now we have these clear streaks that it sort of can all be aligned in time, these upticks. And when we then take the average with this aligned time, so now this is no longer aligned to the saccade, this is sort of an arbitrary realignment of the data. You can see that the two profiles are really similar. It's just that SC activity is higher after we inactivate it. Sorry, that LIP activity is higher after we inactivate SC. And what that tells us is that the upticks are still occurring in LIP after SC inactivation, but their correlation with decision termination is blunted. So it's as if LIP is trying to tell SC that you should end the decision. It's giving the stop signal, but the message isn't going through. And so nothing happens, the integration still occurs, the decision's prolonged and more evidence gets accumulated. Eventually enough is accumulated to actually generate the saccade. So that's the idea behind that. So going back to our hypothesis, which was that SC implements the decision threshold manifesting as bursts, I showed you that SC inactivation impairs this threshold mechanism such that more evidence must be accumulated in order to make a contralateral choice. So my last slide, I want to walk through what do we actually think is going on, right? Because things are more complicated than just LIP and SC. There's more than two brain areas, like I showed earlier. And we think that in particular, the substantia nigra pars reticulata is playing a really important role in this. Now, SNR supplies tonic inhibition to SC, and this tonic inhibition is released right before an eye movement. And the idea is that this release or the suppression of inhibition from SNR generates a burst in SC, which then moves the eyes. And so we think that this tonic inhibition is playing an important role in this threshold mechanism. And if we start with just sort of two assumptions, then we think we can have a clearer picture of what's going on. The first assumption is that upticks in LIP are sort of tickling the bursting mechanism in SC, that when an uptick in LIP occurs, you're right, that you're triggering or making it more likely that a burst will occur in SC. And the second assumption is that SNR activity is negatively correlated with LIP activity. And so with these two assumptions, I'll walk through an example trial or a simulated trial of what might be happening. And so early on in the decision, accumulated evidence is low. And so LIP activity is low, SNR activity is high, right? SC is doing its thing. And early in the trial, there's an uptick in LIP activity. It sort of tickles the bursting mechanism in SC, but because SNR activity is still high, burst in SC is quickly suppressed, and the decision continues. Now, as the decision continues, more evidence is accumulated, LIP activity increases, SNR activity decreases, and now when this similar uptick occurs in LIP, SC is released from inhibition, and it's sort of unleashed to fire a much larger burst, which can actually generate a saccade. The eyes move, and integration in LIP is terminated. So this is a hypothesis. We'll have to test this, but the main prediction is that SNR activity should be negatively correlated with LIP activity. And you might notice that I'm not including FEF in this plot, because the idea there is that people have still not looked at single trial activity in FEF, right? As I showed you, the trial average and the single trial activity can look very different. And so it still remains to be seen whether FEF activity looks more like SC or looks more like LIP, or maybe looks something more in between the two. So I'll end there and I'll summarize. I showed you that single trial dynamics in SC are qualitatively different from those in LIP. And I think this is just a really important point for a lot of systems neuroscience in general, is that if we had just looked at the trial average activity, we would have reached different conclusions, right? The trial average activity was hiding this difference in dynamics in the two areas. I showed you that SC bursts are consistent with a threshold computation, are predicted by a distinct signal in LIP, sort of involving the derivative in LIP activity. And finally, I showed you that SC inactivation impairs the threshold mechanism, requiring more evidence be accumulated to commit to a decision. So to end, I'll say that, to sort of make this relevant to people who care more about the cerebellum or not necessarily decision-making, I think systems neuroscience in general has focused a lot on how the brain processes information or represents things or does a process. And here we're asking a question about how does the brain terminate that process? How does the brain decide, I'm not going to do this, I'm going to do that now? And I think that is also a sort of fundamental thing that the brain needs to figure out. And you can imagine that deficiencies in stopping the process could contribute to a number of mental disorders. So with this burst of activity, I'll terminate my talk and thank the people who helped with the work, namely my co-authors, Eric Troutman, who's critical of getting the neuropixels probes up and running, Danique Jerison, who helped a lot with the inactivation experiments, and of course Mike, who mainly played guitar the whole time. And with that, I'll end and I'll take any questions.
Gabriel, thank you so much. That was just wonderful. Let me start by, I guess, first I thought that the work that you showed with regard to the inactivation made me think about Rich Krause's work on the inactivation of the colliculus, but I thought you so nicely showed that it's not just that you are not paying attention to the evidence, because you showed that the slope was actually greater after inactivation. But I didn't quite understand the model at the end, and trying to make sense of why single trial dynamics differ in Colliculus from LIP. Because here, what you show is that the activity in SC is just showing on individual trials these little bursts. So why is it that that increased excitation from LIP and reduced inhibition from SNR isn't producing a rise in the SC activity?
Yeah, I'm not gonna have a good answer for you. We were shocked when we saw this, and I will say that there is a slight. It's not just bursting that's happening in SC. That there is, if you take out the bursts, there definitely is still a slight increase in activity in SC. So this is probably oversimplified, this schematic, but yeah, I think it does seem though that SC sort of hits a ceiling before the saccadic burst occurs for some reason, and how that's achieved through neural circuitry is an interesting question. We definitely want to test this idea in sort of spiking neural circuit models. Yeah.
Rich, you have your hand up.
Yeah. Thanks for the talk. And I really like that difference between the average data and the single trial. It does seem, though, on this figure that it changes your story, I thought, because it's really saying the thresholds being implemented by the substantia nigra in this case, it seems like. I mean, it makes, the interesting prediction that you get ramping activity there in the opposite direction, right? Gradually decreasing. But aren't you really saying that, basically the colliculus gets this direct input that's kind of priming the eye movement, but unless you have a sufficient inhibition removed from the nigra, you don't get the release of that saccade. So to me, that sounds more like the threshold gets moved into the substantia nigra, not into the colliculus.
Yeah, I mean, I think like the idea that we're going to put like a specific part of the model to a single brain area is probably not going to be successful in the end. I think the way to think about it is that the threshold is implemented with all these regions cooperating, right? You need the bursting, right, that senses a critical level or some event, right? You need the inhibition from nigra, and I think together the way to think about it is that those things all interacting together implement the threshold. But yeah, I mean, I think conceptually, I totally agree with you.
I see. So the colliculus, you can think of it's basically responding to the derivative there, and that derivative has to be large enough for, on top of a baseline, maybe, for you to actually get the system to start making an eye movement in a sense.
Right, or in other words, that the LIP, I mean, again, this is all like mainly speculation, but the idea is that maybe what LIP is sort of representing the evidence for the proposition of the eye movement, right? And that it takes something else to actually implement it. That you're sort of priming, like you said, to use your language, right? Priming the ocular motor system to make, or biasing the ocular motor system to make a particular saccade. That's what LIP is doing.
Thank you.
Debopriya, you had a question.
Yeah, so it was a very nice talk, but while you showed the saccadic and the non-saccadic burst, it reminds me that there was this paper from Annie Churchill's lab back in 2020 where she talked about how due to multiple trial averages, these small burst-like activities might get lost, but they are caught in the single trials, and mostly they occur due to uninstructed movement. So since you suggested that these non-saccadic bursts are kind of not aligned, so do you think that uninstructed movements could be the origin of these non-saccadic bursts?
Yeah, so a version of this question gets asked basically every single time I give the talk. And so I think that the most obvious question is, well, are these non-saccadic bursts occurring when there are microsaccades? And I think that's probably what you're getting at. And the answer is no. So here I'm showing you the instantaneous eye speed aligned to the non-saccadic burst. So that's that black line, and the gray line is if we just do a shuffled alignment. And so there seems to be no, at least in the eyes, there's no eye movement associated with the non-saccadic bursts. So yeah, so it's not explained by eye movements. I mean, with the other, spontaneous movements like fidgeting, things like that. But we didn't have a camera, but given Colliculus, I'd be really, I'd be pretty surprised. But it's possible.
I see. Thanks.
Jorge, you had a question.
Yeah, hi. Great talk. So it's a question also about these non-saccadic bursts, and wondering if instead of substantia nigra, or maybe on top of it, the rostral activity on the colliculus could also be different. And maybe in trials where the rostral activity is high, that burst is not enough to overcome the fixation.
Sure. Yeah, I think that's probably going to be true as well. I think all of these things are correlated with each other. Yeah, I mean, what the causal structure is I think a little harder to work out. Yeah, but that I definitely think in the rostral colliculus we'd see similar dynamics as well to the nigral activity.
Any other questions for Gabriel?
I had one other was, does the firing rate of the LIP, is it lower for the opposite direction movement when you do the inactivation? That is, does its threshold level still remain the same, or does it drop to some evidence of competition between the two? Or are they independent?
Right. Yeah. Good question. I don't have a figure on hand for that, but on the opposite choices we see basically no change. And I only showed you activity at the end of the decision, right? Where I showed this difference on the left. If you look at the beginning of the decision, there's very little effect. And interestingly, I know this wasn't exactly your question, but one concern, right, is how do you know this isn't just a general gain increase in LIP, right? It's sort of trivial gain increase. And what we also found, which I didn't show, is that the visual transient in LIP is actually decreased after SC inactivation. So, all right, so that suggests we're not just simply increasing the gain in LIP by inactivating SC.
Yeah, related to that, but like I was thinking the same, but I saw the reaction time data. I think it was convincing also that there was no decrease on reaction time on the opposite side. Right.
Right. That's
yeah.
Right. Yeah, that too. Yeah, we were pretty surprised how specific and sort of clean the effect was. We had this we really did have this prediction going into it, and Mike and I talked about it, and we thought this is what we predict, but there's no way this is actually going to be what we find. And it was to our surprise, seemed to be just that. Yeah, it's always nice.
Thank you so much, Gabriel. Thank you, everyone. Any final questions for Gabriel? All right. Great to be with you. Have a wonderful Tuesday. Bye, everyone.
Thanks, everyone. Take care.